In case 3, a mother presents her child to the doctor with symptoms of lethargy, polydipsia, which is increased thirst, and polyuria, which is increased urine output. Upon looking at blood samples from the child and comparing those to that of a control group, we notice lower serum insulin levels in the child in both the fasting state and after a glucose loading test. This overly simplified diagram shows the relationship between blood glucose and the pancreas in maintaining homeostatic levels of blood glucose. In the fasting state, low blood glucose levels signal to the pancreas to release glucagon, which then acts upon the liver by beginning the process of glycogenolysis and gluconeogenesis. The most important product of both of these pathways in this case being the release of free glucose into the bloodstream to maintain normal blood glucose levels. So while there is lower than normal serum insulin levels during the fasting state of our case patient, it is not until we look at the fed state that we will truly understand what is causing this patient's symptoms. In a glucose loading test, free glucose is absorbed into the bloodstream and will act upon the pancreas by causing a release of insulin to act as a chemical messenger to various tissues to absorb glucose and use it in storage processes. The fact that our case patient's serum insulin levels are significantly lower than that of the control group indicates that the pancreas is not generating enough insulin to respond to elevated blood glucose levels. This is consistent with diabetes mellitus type 1, which is an autoimmune disorder in which the body attacks the beta cells of the islets of Langerhans in the pancreas, which produce insulin. Without insulin present, the process of glycogen synthesis in both the liver and the skeletal muscle will be interrupted. However, the glucose transport protein GLUT2 present in the liver is not insulin dependent, and therefore glucose can still be transported from the bloodstream. GLUT4, on the other hand, which is present in both skeletal muscle and adipose tissue, require the insulin binding receptors to be activated in order for a conformational change to take place, which will allow the glucose transport protein GLUT4 to be in a position to uptake glucose from the bloodstream. Without the process of glycogen synthesis, the body will continue to produce glucose via glycogenolysis and gluconeogenesis. Therefore, we should see an increased blood glucose level in our case patient when compared to the controls. Without insulin present in the body, cyclic AMP will not be inhibited. This will activate cyclic AMP-dependent protein kinase, which will phosphorylate and activate fructose 2,6-phosphatase and inactivate and phosphorylase phosphofructokinase 2. This activated regulatory enzyme will act as a protein phosphatase and dephosphorylate fructose 2,6-bisphosphate into fructose 6-phosphate, which will be used in gluconeogenesis and converted to glucose 6-phosphate. And once acted upon by glucose 6-phosphatase, it will be dephosphorylated to glucose. When considering glycogenolysis, a lack of insulin will not inhibit cyclic AMP. In fact, a presence of glucagon will also activate cyclic AMP. Cyclic AMP will phosphorylate the regulatory enzyme glycogen phosphorylase and activate it, while also phosphorylating glycogen synthase and deactivating it. Glycogen phosphorylase will free up glucose 1-phosphate from glycogen. It will become isomerized and converted to glucose in the liver by glucose 6-phosphatase. With deactivated glycogen synthase, glycogenolysis will continue unhindered by glycogen synthesis. In case 4, a 2-month-old is presented with jittery spells that occur after breastfeeding. The child's diet has solely consisted of breast milk to this point. The blood work shows that the child is hypoglycemic in the fasting state, meaning having a low blood sugar. Other important findings include hepatomegaly and enlarged liver. An easy rule out here is hereditary fructose intolerance. Fructose is a simple sugar found in fruits, often in the form of sucrose, which is a disaccharide of fructose and glucose. In a normal person, fructose is ingested and phosphorylated by fructokinase. Fructose 1-phosphate is then converted into products that can ultimately enter the glycolytical pathway by aldolase B. Individuals with hereditary fructose intolerance lack the ability to produce and use aldolase B, which leads to an increase of fructose 1-phosphate, which will accumulate in the liver. Interestingly enough, hereditary fructose intolerance presents with hepatomegaly and fasting hypoglycemia due to inhibition of the processes of glycogenolysis and gluconeogenesis due to a lack of free phosphate groups. However, having not been exposed to fructose in the diet, A is likely not correct. Galactose is a simple sugar that is found in milk, so our case individual will have had exposure to galactose in their breast milk diet. Galactose is phosphorylated to galactose 1-phosphate by galactokinase. Individuals with galactosemia lack the ability to utilize GALT, which is the enzyme that converts galactose 1-phosphate to UDP glucose, which can then be used for glycogen synthesis. Galactosemia leads to the accumulation of galactose 1-phosphate in the liver, which is particularly toxic and therefore part of the newborn screening process. If not screened for and does occur, infants can experience cataracts, lethargy, and potentially fatal effects of the liver toxicity. Glucose 6-phosphate dehydrogenase is a regulatory enzyme of the pentose phosphate pathway. Individuals with glucose 6-phosphate dehydrogenase deficiency lack the ability to convert glucose 6-phosphate into 6-phosphoglucolactone. This step in the pentose phosphate pathway is important as it generates NADPH through a redox reaction. This deficiency will also slow down the production of ribose 5-phosphate due to lack of reactants, but will not entirely inhibit it, as ribose 5-phosphate is also able to be produced independently in the non-oxidative phase of the pentose phosphate pathway. Without the ability to regenerate NADPH, the body will not be able to facilitate the reduction of glutathione via glutathione reductase. Glutathione is used by the body to detoxify reactive oxygen species, including hydrogen peroxide, which is damaging to red blood cells. Individuals with this condition would present with anemia due to a lack of healthy red blood cell count. That leaves us with glycogen storage disease. From here, it is important to understand the role of the epinephrine tolerance test that was conducted on our case individual. Epinephrine, simple to glucagon in the fasting state, activates cyclic AMP, which is used in the glycogen synthesis glycogenolysis pathway. Cyclic AMP phosphorylates and deactivates glycogen synthase and phosphorylates and activates glycogen phosphorylase. These phosphorylations lead to the trend towards glycogenolysis, leading to a release of free glucose into the bloodstream. There are several different forms of glycogen storage disease, which affect the body's ability to produce enzymes, from glucose 6-phosphatase to glycogen branching or debranching enzymes. Our case individual most likely suffers from von Gurk's disease, or glycogen storage disease type 1. This disease prevents the activity of glucose 6-phosphatase, which dephosphorylates glucose 6-phosphate to glucose. This has the effect of increased glycogen stores in the liver, leading to hepatomegaly. The fasting hypoglycemia is logical due to the inability of the body to complete the glycogenolytic and gluconeogenic pathways, with elevated levels of glucose 6-phosphate present. Glycolysis will continue to act upon this reactant to produce pyruvate. Pyruvate will continue to be converted to lactate by lactate dehydrogenase. This buildup of lactate in the blood can eventually lead to lactic acidosis and have severe damaging effects on the body due to continued decrease in blood pH. Therefore, the answer is most likely C, glycogen storage disease.